is the day in which me and my dog are going to stop crime in the neighborhood. My name is Mike, and I work with a special command of the police that is in charge of the neighbor security. Recently, there has been a lot of stolen house because of this guy. He has been escaping away from me and my dog since last month, but today I have a secret plan. I'm going to catch him while he is in the neighborhood.
There he is! There he is! Hey, police! Stop! Oh, shit! Oh. Hey, help! Oh. Oh no, I have to take her to the doctor.
Hello, what do you need?
Help, I need help. My dog is dying and I...
Follow.
Yeah, you have to make something before he dies to me. You have to make something for me before he dies.
Let me take the dog first.
Thank you.
Okay, follow.
Okay.
Take seat, please.
You have to make something very important to me. I need you to transform me genetically.
Okay, there are some things you need to know before I make this procedure.
Okay, what are them?
First, the DNA of a human is very similar to the DNA of a dog, so it is possible to make a transportation of DNA from this animal to the humans.
What else?
Second, human smell has a problem. The olfactory receptor gene family is in a 60% of pseudogene, and this means that they don't work. And we have lost that because we learn more things like color vision, which means we couldn't use that at all.
Okay, so you can help me?
Yes, I just need DNA from the dog and to put it inside. Need to take the DNA from the dog. Now we need to separate the DNA. Okay, so to do this, we're going to make the following process. First, we're going to take some DNA out of the dog and we cut it in pieces. So now to explain this better, I'm going to show you what's happening inside. Okay, inside we have some DNA of the dog that is show us how it's developed and how it looks like all in this little thing that is the genome. Now there is a piece of DNA in this genome that has the olfactory receptor genes that the dog uses to survive. Our goal is to make copies of these genes and put these genes inside you. So that way you have more olfactory receptor genes and can have a better smell. To take out this gene in specific, we need to use restriction enzymes to cut this gene in specific places where only the restriction enzyme we are using can cut. Today we are using the restriction enzyme Hey3 that can cut the olfactory receptor genes in certain places. When we cut this piece, but there is a problem, and it is that all the pieces that the enzymes could cut were cut. But it's okay because we can choose which one we want later. Now after this, we now get the plasmid, which is a circular ring of DNA that can exist in a bacteria, and have some traits that can change the way the bacteria acts or looks. Now to add the dog DNA into the plasmid, we need to open the plasmid in two. So we use our HAE3 restriction enzymes in the plasmid and then open it. When we open the plasmid, we insert the DNA of the dog and allow the pieces to join the cut plasmid. So basically, we have some pieces of DNA and some cut plasmids. I need to add the olfactory receptor gene into the plasmid, and the way we are doing it is by adding DNA ligase, which works like a tape, combining the DNA with the plasmid. Before we continue, I have to tell you that there is a chance that this doesn't work, because the way our restriction enzyme cuts the DNA doesn't leave a sticky end, but instead it leaves a blunt end, which is harder to combine with other DNA, although it's possible. Okay, let's continue. There is something important about the plasmid, and it is that it carries a marker that is resistant to some kind of antibiotic. Anyways, after combining the DNA, we're going to get some samples that don't match the gene that we want to copy, apart from the one we want to copy. Some may have different genome, and others may not even get any dog genome. Now that we have the plasmids, we're going to put it into some bacteria. When we take the bacteria, we get some DNA that is already inside the bacteria, but it isn't really easy to make the plasmid enter the bacteria. To do that, we need to cool all the cells down and then quickly heat them up, which will create little holes in the cell membrane. Then the plasmids could slip through and get into the cell. After that, the holes will close up. Now we have the bacteria we want that includes the plasmids with the gene we want. But also there are other ones that have other plasmids that we didn't want, or that don't even have a plasmid at all. After that, we put the bacteria into an antibiotic, and there the bacteria that had no plasmid weren't able to grow, but the other ones will. After that, we incubate the bacteria during a while, letting the bacteria to grow. When we take the bacteria out, we have a problem in identifying which bacteria has the genome of the olfactory receptor of the dog that can be placed inside humans. What we do next is that we separate the plasmid from the bacteria again. We may notice that genes of the DNA we want and of the one we don't want have different sizes. So to identify which one is which, we use a gel box and use the technique of gel electrophoresis. And what this will do is separate the DNA pieces out, and that way we can identify which is the cell we need to copy. We copy that cell many times, and then after a while we insert them on you. Now I'm going to put you the bacteria with the DNA inside. Probably you will feel the effects later, but that is it for today.
Okay. Don't die! Please! Don't die! No! Don't die!
The police and whoever's helped him is going to die.
I'm destroyed. Now that my dog died, I just hope that what the doctor did to me worked. I'm feeling weird inside, like if my body started to work differently. I think that now I'm starting to look in black and white. Probably it's an effect of the insertion of the bacteria. And well, dogs are like that. Anyways, I will do this and more just for him, my best friend, my dog.
It's not over yet.
Now that I have my super snow, I can have the revenge of my dog. I will find you. I can smell him. Yeah. Hey. 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 That's for my dog.
You have to make something very important to me. I need you to transform me.
¿Cómo hago, perro?
Don't die, please.
Oh no!